Okay, I don't want to just be standing around in silence for the next few minutes, so I think we've got a good number of people already joined in. If anyone has any issues, then if you can just drop a line to myself or via the question tab, or even an email if you're really struggling, and I'll do my best to let you in. As I said before, this is our 30th webinar looking at microbiology tools in bioremediation. And today we've got Jude Huggan from NCIMB, and probably everyone knows Tom Aspray from multiple other talks in the past, and currently at ERS. I will just, as I said in the chat, if anyone has any questions, if they can put the questions into the questions tab, and if you can address who your question is for, that'd be great. And we'll have questions being asked at the very end of both talks. I think that's all the housekeeping, and I will pass it over to Jude to do our introductions. Thank you.
Thanks, Stephen. I'm just going to share my screen now. Great. Well, good afternoon, everyone. My name is Jude Huggan, and I'm Business Development Manager at NCIMB. Delighted to be able to participate in this webinar this afternoon to tell you about microbiological tools in bioremediation. Firstly, I'd like to just run a quick poll. So, Stephen, could you run the quick first poll for me, please? Hopefully that questions should appear on your screen.
Yep, that's
okay, so great. So I just really wanted to gauge your level of understanding. So question one really is, what is your level of awareness, understanding around the use of molecular tools such as PCR and sequencing in remediation projects? And if you could pick the answer that best applies to you, that would be great. I'll give you a moment or two to answer that question.
Yeah, there's still a few people answering, so I'll give it a couple of seconds more. Okay, just for your, I don't think you're able to see it, Jude, but
no, I see it now.
Okay, so about 40% of people are not aware of it, and 40% have a general understanding but never used, and then only 14 to 14% of people have used it before, and only 2% have a high level of understanding for it.
Okay, no, that's great. So again, just moving on quickly to question two, really as a follow-up question is does your organisation currently use microbiology or molecular tools to investigate soil and groundwater? And that's just a simple yes, no. Again, just for me to gauge your level of understanding.
Yep, I've just started up that poll. Give it a couple of minutes, or a couple of minutes. Okay, can you see that there, Jude? Okay.
I can, yeah. So quite a lot of people don't, but some do. Okay, that's great. So we can move on to you can share, give control back to me. That's great. So in terms of the outline of my talk today, I'm going to introduce you to NCIMB, tell you who we are and what we do. I'm going to provide an overview of our services, highlighting in particular the services that I think are of most interest to this audience, in particular introduce you to the molecular tools that we have to offer. And then I'm going to talk through a couple of case studies highlighting where we've used molecular tools to help customers with their projects. So just a bit of, I guess, background. You know, microorganisms are absolutely everywhere. They're estimated to be around 1 trillion species on Earth, with only 99.99% of, or only with 99% of those yet to be discovered. And over generations, we've harnessed the potential of microorganisms for our own benefit. So if you think about food and drink, we obviously know them in terms of wine, cheese, beer and bread production. And interestingly, recently, they've been used to manufacture alternative proteins such as corn. They're also really useful in terms of manufacturing things like antibiotics. So if you've ever taken amoxicillin for strep throat, that's made via bacterial fermentation. So really we can use microorganisms really as factories of the future to produce things of high value, such as chemicals and vaccines. But on the flip side, there's obviously microorganisms that can do harm, both from a sort of clinical sense and I guess more widely those organisms that are likely to affect product and process quality or pose a risk to consumer health. So who are we as NCIMB? Well, NCIMB stands for the National Collection of Industrial Food and Marine Bacteria. So essentially think of NCIMB as a collection or a library of microorganisms, and for the last 70 years, or over 70 years since 1950, we've been collecting and storing microorganisms and then using those microorganisms, allowing customers to access them for research and development purposes. And over time, we have evolved to become a microbiology, analytical and biological storage organisation based in Aberdeen, really providing services to support organisations who use microorganisms to either develop new products or new processes, and also services to organisations that have the need to monitor for the presence of specific microorganisms or groups of microorganisms across various environmental niches. So for example, within oil and gas, our services are widely used to assess the threat of microbially influenced corrosion. And so this slide just shows the evolution of us as an organisation, and we're very happy to announce that we're moving into custom built premises next to Aberdeen Airport over the next couple of months. So although we've been going as an organisation for 70 years, actually we only started as a limited company relatively recently in our lifetime, 40 years ago, where we support academia and industry, really helping those organisations with their research and development projects, providing products and services to help carry out and comply with quality control procedures. We help companies to safeguard their intellectual property and protect their important biological assets with storage and offsite services, but also helping companies comply with regulations across particular environments. So who do we work with? Well, actually, we work with a variety of different organisations across multiple sectors, and you can just see a snapshot in the boxes on the right-hand side here of the types of sectors that we work with. But broadly speaking, we work with companies who are developing microbial products and processes, so think things like pre and probiotics, think live biotherapeutics in healthcare, animal feed additives, silage inoculants, anything really that has a microbial application. More recently, we've been involved in industrial biotech projects where people are looking for alternative processes that don't involve fossil fuels, but involve much more bio-derived fossil fuels. And actually, that's not just about using microorganisms in products and ingredients, but actually to produce certain products. So think enzymes, flavours and fragrances, and also cosmetics. And increasingly within the sort of environmental and soil health area, microbial bioaugmentation and bioremediation products. We also work with companies that have a need to detect, identify and monitor particular microorganisms, really across environments and niches where microorganisms have the potential to affect product and process quality. So for example, across various manufacturing facilities, such as food and drink production, but also pharmaceutical manufacturing, and in particular highly regulated environments such as clean rooms, it's important to not have any microbial contamination that could affect products that are going into human or animal health. Increasingly, we're being asked to identify communities of microorganisms and monitor those changes, the changes to those communities over time across various environmental niches, such as within water or soil samples, and also across the gut microbiome. So again, assessing the effect or changes within those communities for oil health implications. And again, I talked about our work within oil and gas production. And increasingly, we're seeing more requests for microbial analysis of specific carbon capture, utilisation and storage projects. So our services today are very broad. We do work across multiple sectors, but really I want to talk in more detail about the ones that I think are more relevant to this community. So I'm going to talk about our ecotoxicity testing service, our metagenomics or microbial community analysis service, and qPCR services that really help to detect, identify and quantify microorganisms. Oh, sorry, too trigger happy there. Okay, so in terms of ecotoxicity testing, NCIMB have developed multi-species microbial bioassays as a reliable measure of toxicity. So there are many ways that people assess ecotoxicity, and that includes using higher order testing on species such as algae, crustacea and fish, as well as using microorganisms. And microorganisms are a really good way of testing ecotox because they're fast, they're easy to grow, and there are no ethical issues associated with that compared with higher order testing. Now, normally ecotoxicity testing with microorganisms is carried out with one specific species of Albuvirio fisheri, and that's commonly known in the product Microtox. However, we provide multi-species microbial testing because we believe this brings many benefits over single species testing, not just greater genetic diversity, but also greater specificity. So we've developed two product lines, so Mara, which uses 10 strains of terrestrial, freshwater and marine bacteria and one yeast, and that essentially you're looking at that's a growth assay to measure toxicity. We also have our Lumimara product range, which uses 11 species, 9 marine and 2 freshwater, and that looks at the reduction in luminescence of those luminescent bacteria, so the reduction in light output as a measure of toxicity. And actually that's used, these products are used widely to test a variety of different materials, effluents, chemicals, soils and sediments to assess their toxicity. Like I say, these are provided in a convenient format. You can see the picture above. We provide these organisms freeze-dried in a 96 well plate, so you can buy the kits and test those on site, or you can send us your samples and we can test those at our facility. Like I say, the results from bacterial assays are obtained quickly. There are no ethical issues associated with those compared to higher order tests, and you can use those to monitor changes over time. So we actually use these tests very widely within the oil and gas industry. BASE have recommended these tests to use to produce water within the oil and gas sector, and that was as a result of a big offshore trial we did with that sector. Moving into other sectors, we have recently started a collaboration that looks at developing these types of tests specifically for soil. So we'd be looking to develop this type of model to assess the toxicity of pesticides on key microbial functions and diversity. So essentially developing an assay for soil toxicity using particular microbes. And we find that this ecotoxicity testing service and kit really complements well with our other services that are used by organisations for effluent and produced water testing, such as BOD, COD, pH and suspended solids. So moving now on to the more molecular based services, I want to talk about 16S metagenomics or microbial community analysis. So what is 16S metagenomics? Really that's a really useful molecular tool that we use to assess all bacteria and archaea within a specific sample. So that involves sequencing the 16S gene, and that's a particular gene that is common to all bacteria and archaea. So it's great because you can then use that barcode, if you like, to then pick out all other bacteria and archaea in the sample that have that particular barcode. I know Tom's going to talk about PCR more specifically in the next talk, but broadly speaking, we amplify this 16S gene using PCR, and then that allows us to get species-specific signatures that we can then use to identify all the bacteria within that sample, and indeed complex samples. And we find that that technique is really good because it's fast, it's cost effective, and it's pretty precise in what it's able to provide. And actually recent technology advances have meant that this technique is the costs of this technique are rapidly decreasing, and actually the accuracy is also increasing, which makes it a much more common assay. And one of the benefits of this is that you don't actually have to culture or incubate the bacteria for growth. You can take and analyse samples directly. So metagenomics really will tell you what is in your samples in terms of bacteria and in what proportion they are in relative to one another. So it's a semi-quantitative technique, and as I say, it provides details around the relative abundances of all different taxonomic groups within the sample. So you can see it's great for checking changes between sites, between locations, monitoring changes over time, or actually in response to specific remediation strategies. So you can see the graph on the left-hand side here, just really a mock example. You can see the list of genus, the strains that are in this sample. You can see they don't change particularly much within the samples that are being tested, whereas on the right-hand side you can see that the samples are markedly different between each other, and that's because they might have come from different sites, they might have come from different locations, or they might have been different time points. But you can see that this is the output that you would get from using the metagenomic tool for this. So like I say, we find this technique really useful in measuring changes over specific time points across locations, and actually it's really useful to use both pre and post remediation to monitor the differences within the differences of that particular remediation technology. So moving on to qPCR, normally if you wanted to quantify organisms you would have to culture them, but it's great now that we have this molecular technique that you don't have to do that. And like I said before, Tom's going to discuss these in a bit more detail, but broadly speaking, we can use this technique to obtain rapid quantitative information on microorganisms, and including the total number of bacteria present in the sample, as well as details of a narrower group of specific organisms or specific genes of interest. So like I said, the technique amplifies a particular gene, makes multiple copies of that very quickly to allow you to do some in-depth sequencing. And of course you will have heard of the technique because it's widely used to analyse and quantify small samples such as forensic-based samples, and of course more recently it's found fame within the COVID testing market because it's been used to detect the presence of disease-causing organisms, in particular COVID. So in terms of total bacteria, what we do is we tell it we want to amplify the 16S gene, so it looks for all the genes that have the 16S marker. You can amplify them to get the total bacteria level. And then if you're looking for specific genes, you have to generate primers, essentially barcodes of that particular gene of interest that are implicated in specific process of interest, and then you can detect and quantify those samples. So those, for example, could be things like sulfate reducing bacteria in an oil and gas reservoir, or hydrocarbon degrading bacteria within specific environmental samples. Indeed, really, if you have any functional genes that you know are implicated in degradation, you can set up assays to monitor for those changes. So you can look for other polycyclic aromatic hydrocarbons or petroleum products or chlorinated solvents, for example, to detect changes and quantify the contaminants of interest. So I want to just talk quickly through a couple of case studies where we've used these to good effect. So case study one really was a customer who was interested in specific bioremediation strategies of runoff waste and how far the runoff waste penetrated across a particular site. That customer was looking to build a new motorway, but that happened to be on an old industrial manufacturing site, and so they were looking at various technologies to determine what was in specific sites, and so that to understand what was the most appropriate remediation strategies to take. So we looked at soil samples from different parts of the site across different location depths in terms of soil, and what we were looking for there, really, again, thinking about 16S metagenomics and qPCR, was what was actually in there, what species richness was there within the sample, as in what did the taxonomy look like, but also how well spread these were across different sample time points. So looking at both the diversity within samples and also the taxonomy, how abundant they were within specific locations. So what we were able to tell that customer was that actually, broadly speaking, all the samples that we tested, the taxonomy were broadly similar. So the top 20 genera involved or highlighted in that particular sample were vaguely similar, but they had the presence of microbes associated with soil, with sulfur metabolism and various organic matter decomposition. And specifically, we weren't able to find any enrichment for species that were involved in hydrocarbon metabolism or indeed other pollutants. And really what that then allowed the company to do was to carry out some additional testing, really, and use this test in conjunction with others to really carry out the risk assessment and understand what the most appropriate remediation strategy was. So again, it's perhaps not a standalone tool, but it's useful to complement other existing tests that we have to remediate or when choosing which remediation products to use. Second case study was really an environmental case study in Africa. That customer was looking to assess particular sediment and water samples for the presence of hydrocarbon degraders across specific locations. And again, we looked at those samples and were able to tell the customer the diversity within those samples, as in how many, the level of species richness and evenness within that sample, and also the most abundant species that were present. So in this case, we were able to say, well, actually, the samples varied very widely in the proportion of genera that we found that were implicated in hydrocarbon degradation, and that was particularly predominant in sediment samples compared to the water samples. We were able to tell them that there were high levels of particular species associated with marine oil pollution, and also high presence of organisms associated with biodegradation of particular environmental contaminants. And then we could take qPCR, having done the 16S metagenomics, qPCR to then quantify this particular genes involved in both marine oil pollution and of those particular environmental contaminants. So again, that just allowed that customer to have a better picture or understanding of that community that they were sampling, and understand what remediation strategies they had to employ, because they were likely to be different across different sites, depending on what was found. So in terms of applications of this technology for LCF members, I hope I've given you an introduction to the technologies and an insight into how they can be used, both in terms of ecotoxicity testing, but also molecular tools that are used to support the identification and detection and quantification of particular organisms in bioremediation. NCIMB really provides the data and the analysis for you as an organisation or organisations to make informed decisions about the appropriate bioremediation strategies to use. We don't sell bio-augmentation or bioremediation products, but we really provide you with the data that allows you, as I say, to make informed decisions around what the most appropriate strategy is to use. So if you have sites that you're looking to assess for particular microbiology aspects, or that require a more comprehensive assessment of sites, in addition to the tools that you're already using either for current or future projects, we'd be really happy to have more of an in-depth discussion on how we can support. Stephen, would you mind putting on the second poll, please?
Yep, I'm just doing that just now. So
great. So thanks for that. Having sort of heard a bit more about the tools on offer, would you consider using microbiology or molecular biology tools for future projects? And if you can answer yes or no.
just getting the responses in now, so it'll just be just a wee second until got a few more. Okay, can you see that okay?
Yeah, so okay, great. So quite a lot of you are interested in considering that. That's fantastic. So I guess maybe my question's more for the small percentage of you who answered no to question 3. But if you did answer no to question 3, what are the perceived barriers to using this tool? So if you use the next question, please. As I say, what are the other perceived barriers that are stopping you perhaps adopting this tool for future projects? And if you could highlight all of the answers that apply. Great, so quite a varied response from all the audience. No, that's great. If you could go back to my screen, thanks, Stephen. Great. So yeah, thanks very much for your attention. As I say, I was really delighted to be able to talk to you today. My details are on the screen. I'm sure the slides will be shared with the community after this presentation, but if you have any questions, I'm happy to answer them at the end of this presentation. Equally happy if you would like to follow up, my contact details are there. Thanks very much.
Okay, thank you very much. And yes, if people